And today we are very happy to have a second speaker in our seminar series that started 2 weeks ago with Manuel Tupu, and today we have Marcin Szwed, who I met 13 years ago now, I suppose, in Trieste when he was a student and I was a tutor. Marcin did his PhD at the Weizmann Institute, focusing on the systems neurobiology of sensory signals transduction. And then he followed that with a postdoctoral position at the Atomic Energy Commission near Paris, working in the lab of Stanislas Diem and collaborating with Laura and Cohen, focusing on imaging techniques of language processing. And now Marcin is associate professor at the Department of Psychology at the Jagiellonian University in Krakow, which is a really beautiful place and a really historically interesting university. And we're happy to have.
So I'll start with thanking Anna, Vasily, and Boris for inviting me. So I will speak about neural systems for reading in the sighted and in the blind. I use mostly fMRI, but also other imaging techniques to study this problem. And I will start by acknowledging all the people who contributed to work I'll be showing today. So when I saw the light and started studying humans instead of rats, I did not know a lot how to study humans. And Emily Kiawa was a master's student at the time. I was already a postdoc, but she taught me all the techniques necessary. Evelyn Egger was always advising me a lot about experimental techniques and designing experiments. During my postdoc, I had the pleasure to have two, not one, excellent supervisors, Laurent Cohen and Stanislas Dehaene. Then in 2011, I worked in Marseille for half a year with Jonathan Granger, and some of the data I'm showing you are a result of collaborations with a group of Jean-Philippe Lachaud in the Epilepsy Center in Grenoble and Lyon, and with Carlos Haname. And the other very fruitful collaboration I had was with the group of Amir Hamedi in the Hebrew University in Jerusalem and his student Leo Reich. So why study the cognitive neuroscience of reading? Well, the reasons are philosophical, because writing is a uniquely human capacity. So it's about understanding our human condition, understanding the crisis of culture in human brain function. But there are also more cognitive reasons why to study reading specifically, because reading is a great cultural experiment undertaken by society. We are not born with reading like we are born with language. It takes effort, it takes educational systems to acquire the capacity to read. So that great learning experiment, which is reading, can provide answers to some of the most important questions about cognition and the human brain. So I'll start with a couple of facts about the acquisition of reading. So you know all it takes time to learn to read, and you can see that when you look at the reading speed in children who are 7, 8, 9, 10, 11 years old, you see that the reading speed, as measured by words read per minute, does not go all the way up in second or even third grade, but it continues laboriously until the 6th or 7th grade. So this learning to read is a long process, and by the end of it, reading becomes effortless, very fast. We all read about 150 to 200 words per minute, and massively parallel. And what I mean by massively parallel, if I flash you a 4-letter word, and if I flash you an 8-letter word, see how much time it takes for you to recognize that word, to read that word. You will see that for letters for words up to 8 letters, it takes almost the same amount of time. And again, it's not something you are born with. You can see that first grade children and also adults like me who are trying to read Russian, they read letter by letter. I know a bit Russian, but I need to read every letter and put it together. So that's what happens in first graders when they're trying to master any alphabet. But by the second and third grade, people get faster and faster, but that's something that, for example, dyslexic children cannot achieve. So what I'm asking currently in my research group, what I've been asking for a couple of years, is how this massive cultural experiment can inform our view of sensory processing. And to this, we have to get to our standard model of how sensory processing is actually being done. So the current view is that sensory processing is done step by step. So it's not happening all at once in one place in the brain, but there are several distinct brain regions that, in case of vision, for example, extract simple feature, then pass it on to another region of the brain, and so on and so on. And at the end, you get to a high-level representation, of specific objects. So in the beginning, at the bottom of the system, you have detection of simple features, and as shown in the work of Huber and Wiesel in the 60s and 70s. But at the upper level, you will have dedicated brain regions that recognize specific classes of visual objects. So you will have a region for faces. Some people even argue that you have a so-called grandmother cells that recognize your grandmother at the level of specific neurons. And you will have brain areas that are more specific to places and landscapes for orientation. So if we take this classical model and apply it to reading, then what we get is a model where, again, words are assembled step by step, going on from simple traits, then through parts of letters, entire letters, to a representation of entire words. And that would mean basically that reading systems at the bottom share the same machinery, the same elements, but at the top you would have a dedicated reading system in the brain that is put in place as you learn to read. So I'm basically after 3 main research questions. The first question is, okay, you have face areas, you have place areas in the brain, but can you have a special visual area for capacity that is a recent cultural invention? Reading has been around for 5,000 years only. That's not enough time to evolve in a biological sense, a dedicated brain center for that. And anyway, most of the population during those 5,000 years did not know how to read. So that's the first question. Can you have a specialized visual area for something that is recent, that is culturally acquired? Second question is this really all that's happening at the first level? Is the function of those primary visual areas really constant, regardless if you're looking at objects, if you're looking at novel abstract things, or if you're looking at words? Or is there a possibility that because you learn to read so much, because that's the single thing that you practice the most in school, some changes might have occurred here at the very bottom level of the processing hierarchy? And the third question we're asking actually goes further than vision. Well, so far, I've been equating reading to vision because most of us read with our eyes. And we know that sensory processing is confined to dedicated brain modules. So, for example, vision has its own brain area, and audition has another brain area. and sensation, tactile sensation has another brain area. But we can actually test this idea that different senses have their own dedicated brain regions by contrasting braille reading, tactile reading, to visual reading. And we can actually put this idea of one modality, one brain region to a test using reading as a research tool. So this is the summary of what I'm going to talk about, and we're going to start with a special visual area for reading. Is there one? What does it mean specialized? So reading has many phases, and it's about recognizing graphical shapes and connecting them to their sound and to their meaning. And naturally, spoken language and written language share many components, and you can see that if you look, for example, at what's happening in the brain when you hear a word and you see a word and you take a snapshot of the brain with MEG about 300 milliseconds after you heard or seen a word. Well, here at 300 milliseconds you can actually see that the brain regions activated are very similar for the spoken words and for written words. But if you go back about 150 milliseconds in time, you will see differences. You will see that spoken words, just after you hear them, activate areas around the auditory cortex, whereas words that you read activate massively primary visual cortex and high-level visual cortex. So that gives us one clue that this part of the brain might be specialized for reading in some way. Another clues of where is the reading area in the brain come from neuropsychology. So we know that cerebroscope accidents in some of brain regions lead to deficits that affect both spoken and written language comprehension in case of Broca's area, and also production in different ways. But we know that lesions to that ventral brain area, which we see activated very early on for reading, lead to something that's called pure alexia. So in pure alexia, word reading is severely impaired, but you can still recognize faces, you can recognize objects, and most importantly, you understand spoken language without problems, and you can even write. So this is another clue where the reading area in the brain might be. And finally, we have neuroimaging. That's what I'm mostly dealing with. So if you take a cartoon of the visual system with, again, simple things being done here, and then in the diverging in two pathways, the dorsal pathway, which is mostly about where things are, and movement and position in space, and the ventral pathway, which ultimately leads to those special areas that recognize faces and objects and so on, you will see that just next to this area that is dedicated to face processing, there is an area that is consistently activated by reading in a number of studies. You can read letter strings, words, and so on. So that area has been called the visual wortum area, and you can see very clearly that it's lying just next to other brain areas, specialized mostly for objects, for faces, and for places and landscapes. And moreover, you can actually see that this reading area takes over a part of the object recognition area. This is best seen, for example, by contrasting people who are illiterate. Okay, so here you have different groups of people starting from illiterates to reading people. You can see that illiterates cannot read at all, and we're talking about adults. This is a group of adults illiterates and ex-illiterates who don't really know how to read, up to very illiterate people. And if you do a brain imaging study on all those subjects, and that has been done by the hand group 3 years ago, if you take a section through that ventral mosaic, through that brain regions that are dedicated to various tasks, and you look what is activated for houses, and tools and letter strings, you will see that in illiterates, there's basically no activation for letter strings, okay? There's nothing almost that's green in that snapshot. But if you take literate people, you will see a patch of green appearing. That's the region that now dealing with words. And you can see very precisely that this region was, at the beginning, an object recognition region. So what we're doing is we're taking our object recognition capacity and converting it to reading. This is called neurorecycling by some of us. Now, how specialized is this region? Well, we might begin the question by looking at how specialized can you get in that part of the brain, and the answer is you can get very specialized. Actually, the classical example, the face area, is built of cells that respond almost entirely to faces. Now, this is a study in macaque monkeys by the Sauer group. What the group does, they look for these face patches in the brain with MRI, and then they go in and record their individual cells with electrodes. And here, what you'll see is activity of 180 cells. So you see each red dot is a spike of activity, and you will see that these cells basically respond almost totally to faces. They will not respond to any other stimuli known to drive the ventral stream. You might say, okay, if there's something inborn in the higher vision, it's faces. We're primates. Primates for millions of years have been relying on face recognition because they're social animals. But reading, as I said, is completely different. It's very recent. So this area starts as an object recognition area, and some actually argue that there's no reading region in the brain at all. That basically what you have is a multifunctional region dealing with both reading and object recognition. And this is a debate that has been going on for a while, and it's still going on. And some people argue, as I said, that basically it's the same cells doing different things depending on the context. Now, there has been at least 15 MRI studies on that subject, and some people found no regions that are more active for words than objects if you look closely. And some other people found, yes, maybe there are some regions more active for words than objects. And there are many problems with those studies. The first one is that basically you're trying to find something that's more active for word than a picture, but a picture will naturally have more visual features, will have more detail, more line junctions than a simplified thing like a word. So you are naturally more biased to find something more activity for objects than for words. But I try to address this problem by simplifying basically objects to the point that they resemble words, and also using this kind of low-level controls in one study. So I also did one of those 15 MRI studies, and in my case I did find a region of the brain. This is an MRI image of the comparison between words and objects. So I did find a region that is more active for words and objects, but to tell you the truth, this difference was very relative. So even in the most specific region, the activation for words, which you see here, would be only about 20–30% stronger than for objects. So we thought that we are in a dead end here, that non-invasive neuroimaging basically cannot resolve this issue. Luckily, there are very rare cases where you can use invasive techniques in humans. Now, this is basically in a situation where people have very hard and epilepsy that cannot be treated with drugs. What you do then, you want to operate on the epileptic focus, but you don't want to cut out the entire lobe of the brain, because such a person would come out completely impaired from the surgery. So in those cases, you basically implant electrodes in the brain of somebody who is about to undergo surgery, and you hope that these electrodes will help you locate the epileptic focus. Now, I always stress here that the location of the electrodes is exclusively decided by neurosurgeons, and we as scientists, we have no saying where the electrodes should go, and we should not have a saying where they go. Nevertheless, we were lucky. We found 2 patients that had electrodes near the supposed reading area in the brain. That was in an epileptic clinic in Grenoble. And these opportunities allow you much higher spatial resolution than MRI or MEG. And moreover, with MRI, you record the physical signal that is linked to blood and oxygenation, blood flow. And here you record broadband gamma activity, which is very close, which is much closer to real activity of brain cells, which is very closely following the population spiking rate. So those 2 subjects, they sit for a week in the ward, in the hospital. They are waiting for the epileptic attack to happen. They are bored. They happily collaborate with researchers. So we had a simple experiment with them. They saw 7 visual categories: houses, faces, animals, scenes, objects, and consonant strings and pseudo words. And we were looking, okay, will we have specific responses to those pseudo words and consonant strings, or will the picture be like an MRI? So some differences, but relative. So this is the result. And here you see responses to all those variable categories of objects, visuals, objects and faces and animals. And you see here the responses to pseudo words and consonant strings. And basically, people, I did not believe those results first when I saw them, and people who record for 10 years in the visual system say that, with the exception of the face area, such specific responses, such big differences between those 2 categories, which are letter strings, all pseudo words, and all the rest, have never been described before in the human visual system. So here, in those very rare recordings, we have evidence that specialized brain regions for reading exist, and moreover, maybe I'll skip that slide, that they can be achieved through cultural learning. So this shows the advantage of using parallel medical techniques through brain imaging, that the snapshots you get from intracranial recordings might be very lucky, might be incomplete, but they provide you with a completely different view than brain imaging. They actually bring very important complement. Now I'm going to move on to a second topic I've been looking at. I've been looking at the function of primary visual areas. So as I mentioned, the classical view is that primary visual areas recognize line orientation. So they are the first place where you process visual input after the retina, and their job is to like a contrast filter, to fish out any line orientation, any contrast changes from the visual world, and then pass on this to the higher level regions, so simple things. But there are some experiments that should suggest that these early visual areas can actually do more complex things. And actually I'm going to show you one of them. It's an experiment by Mariano Sigman and Charlie Gilbert. Here that Mariano and Charlie, they asked people to detect an inverted T in an array of distractors, T's that are lying on the side. So what you're going to do is you're going to look here and try to tell me if you see inverted T's. Did anyone see inverted? Okay, not really? Okay, there was inverted T, and if we sit here for another 6 days, you're gonna get much better at it. So there's nothing wrong with you, maybe it's the evening, but after 6 days you would have gotten much better. But moreover, after 6 days, if we've done an MRI scan, you would see that the activity for trained Ts, that basically the trained Ts will be processed by early visual areas, whereas the untrained Ts will be still processed by regions of the brain dealing with attention, the so-called attention network, and by ventral regions in the brain. So this is a classical example of perceptual learning. The best example of perceptual learning is, for example, radiologist training. So a radiologist will spot a small pathology on an X-ray very quickly, and you would take, if you're not a radiologist, you would take minutes to look for it serially, and most likely will miss it anyway. And perceptual learning can lead to rapid and parallel detection of complex visual targets. There are many experiments on that. And actually, if you look at reading, reading is very quick and very good, but only when you are presented with reading in familiar orientation. So if you look for a word chair in this array of distractors, that will become very quick for you. But if you look for a word chair in this array of distractors, that will take much longer. And that is also a hallmark of perceptual learning that is very specific. This type of learning is very specific to the stimulus orientation, to where it appears in the visual world. So that would suggest that perhaps what you have in reading is a form of perceptual learning, and perhaps you have a modification of early visual areas in reading as well, and not just the high-level visual from area. So again, I did an experiment here where I tried to select the size of those individual components making my objects and my words in a way that would correspond to what a V1, what an early visual cell sees. So an early visual cell should see a cluster, one element like that. So basically the prediction here, if you take the primary visual cortex, is that those, the responses to those 4 categories of objects should be more or less the same. Well, maybe the scrambled things should give you a bit more response because they have higher frequency. And the result I got was pretty surprising. Basically, we discovered that the expected thing is happening for objects. So we have less activity for entire objects than for scrambled objects, but for words, you have actually more activity than for scrambled words. So this is the reverse of what you would expect. And when we found that, well, we said, well, there's something special happening for letters, for words in early visual areas, and this is an effect of perceptual learning. But the first criticism we got that you're comparing objects to words, and this is really like comparing objects, comparing apples to oranges. So you cannot really tell what's going on there. So this is the next experiment we did. We basically took 2 very different writing systems. We took the Latin script and we took the Chinese script, which is very different visually. And we bet our chances on the fact that this kind of early V1 learning is very specific to the shape of the stimulus you learn. So the prediction was that if you take people who are not expert readers in Latin, namely students that came to Paris from China just 1 or 2 years ago, who are not yet fluent readers in Latin, then you basically should expect that these early visual phenomena for letters will occur only in French readers. They will not occur yet in Chinese readers. So this is the next experiment where we contrasted Chinese participants and French participants. The French participants, when you ask them to name Chinese words, they're at 0%, they're at 100% for French words. Now the Chinese students are of course perfect in Chinese words, and it's not like they cannot read French because they study in French schools, but when you impose a high reading speed of 150 words per minute on them, when you force them to read quickly by flashing words, they actually will break down. They will be about 50% correct. And this is the finding in a nutshell. So what I'm going to show you is activity in the left visual hemispheres for French subjects, Chinese subjects, and the black will be the entire words and the white will be the scrambled words. So first of all, in this experiment we replicated our previous finding, more activity for entire words than scrambled words in French readers. But the nice thing is that this effect is specific to French readers. The Chinese students did not show this effect. And the right hemisphere there was no difference. That's because language is localized to the left hemisphere. So basically what you see here is those enhanced activation for reading. They are predominant in the left hemisphere, but they were not observed in readers who were not yet expert. And the other nice thing is that if you look closely at the area of the visual system, at the area of V1 cortex where they occur, the area of those enhanced activations corresponds to what we call a horizontal meridian. So basically, these enhanced activations happen exactly where you expect to see new letters coming up if you're a reader of the Latin alphabet, if you're reading from left to right. So basically, this is causal evidence that these early visual activations are not just a byproduct of comparing objects to words, but they are really related to visual expertise. Now, okay, now we're 5 years into this research program. We have 2 experiments, but we still don't know, is this just a byproduct of reading or is this really important? And this is basically because we've been using so far only one type of experiments where you have 2 conditions, for example, letters and scrambled letters, and you do a comparison between these 2 conditions. And the next idea we had is to use a different design, is to use a different experimental method in hope to bring a different kind of evidence. So what we did next, and what we're currently analyzing, is an experiment where people see the same thing. They see always a string of letters. And you have to, the task, you ask them to detect, say what's the letter in the middle. But you flash letters so quickly that sometimes they are right and sometimes they are wrong. So here, you use identical conditions and you play on the behavior of the subjects, whether people were correct, whether people were incorrect. You compare activation for the correct recognition trials in the brain and the activation for the incorrect trials in the brain, and you take the brain regions where there's a difference between those 2. So I'm going to skip over the experimental design. Basically, people see those 5-letter strings. They're either letters that are super practice, you've seen 100 million of them in your life, or false fonts that are completely novel. And then they have to decide what was in the beginning, and you make it hard, so sometimes they succeed or not. And these are the false fonts that I was talking about. And this is very preliminary, we're still analyzing that, but just to give you the gist of the result, if you take the higher level regions in the brain and you compare the activity for correct false fonts and incorrect false fonts, you get a difference. And you get the same difference more or less for letters, and these are also letters that are played for a longer time to control for the stimulus duration. But as you move on to early visual areas, you will see that here, in V1 and V2, you have a difference between correctly detected letters and incorrectly detected letters, but you don't have that difference for false fonts. So here is another type of evidence that these early visual areas, they are really overtrained, they really do something about a reading that is specialized. And if you ask me what's really happening there, I must say that we still don't know. It could be the fact that you might do complex things, you might detect entire letters already in your early visual system, and that gives you an advantage, the possibility to read quickly, to read entire words in parallel. But it could be also some kind of feedback mechanisms where the higher levels of the brain feedback to V1 and say we want that high resolution information about the letters to extract their identity. But nevertheless, the conclusion from that part of my talk would be that the impact of literacy on the visual system is much more widespread than expected. And in fact, literacy influences also primary visual areas. So in the last part of my talk, I'm going to focus on braille reading, on reading in the blind. So the amazing thing about this reading area in the brain is that it always happens in the same place across cultures and writing systems. And this is true both for people within people. Let's say there are people like in Japan who treat both syllabic script like kana and logographic script like kanji, which is basically Chinese. So these two scripts will coexist within the same brain regions down to a few millimeters. But this is also true if you compare different subjects from reading different scripts. In the experiments I told you, which is actually published already in Journal of Cognitive Neuroscience, you will see that French reading French and Chinese reading Chinese will activate the same brain regions. But what will happen with Braille? What will decide the location of the reading area of Braille? Is it the modality, the sensory modality, which is completely different? We have touch in one case and vision in the other case. Or is it the task, which is reading, which is the same? And now, this gives us a possibility to question the established dogma of division of labor in the brain by contrasting Braille reading and so-called normal reading. Now, if you were just to look in the textbooks where the blind reading area should be, you would get the same answer from both neuropsychology and from neuroimaging. This is an atlas of brain areas made based on brain wounds in World War I, compiled by Kleist. He put together about 300 cases, and according to this atlas, tactile recognition is completely different place than visual recognition, and therefore reading Braille should be in different place than reading with your eyes. But if you zoom into the 21st century, and this is the most established textbook in our field, you will see that, again, you have this division. The association cortex for vision is one place, the association cortex for tactile reading, for tactile recognition is completely different place, and therefore tactile reading should happen somewhere else than normal reading. But nevertheless, we tried this experiment with Amir Ahmedi's group in Jerusalem. So we took 8 congenitally blind people, so these people have no visual experience at all, and we scanned them while they were reading Braille and also a number of control conditions. And what you're going to see here is activation for Braille reading in a group of 8 blind subjects versus the activation for visual reading in a group of 8 adult normal controls. And you see, to our surprise, and again, I did not believe those results at first, that Braille reading in people who have no visual system activates exactly the same brain area as visual reading in sighted people. And in fact, if you take the peak of reading activity in each person in blind and in sighted, they will fall all in the same neighborhood, and it's enough to remove the color code and you cannot tell who is blind and who is sighted. So what's going on here is that we see that this visual cortex is not completely visual when it comes to reading. You actually have a tactile reading area in the place that's supposed to belong to the visual cortex. Now, why is it there is an open question, and we're trying several experiments to understand what's happening there, but nevertheless, this result we got 2 years ago challenges the canonical view where the brain is strictly divided according to the sensory domains, and suggests an alternative view where the division of labor is more according to the tasks. So if there's a task like putting together different simple stimuli and communicating it to language, which is reading, then you would do it more or less in the same place regardless of the modality. So, to summarize what reading can tell us about sensory processing, first of all, as we've seen, learning to read can lead to the development of specialized brain regions, so strong specialization, at least in the ventral visual system. That's the part of the brain that has a tendency to specialize. Is possible even through cultural learning, so the extent of culture-dependent plasticity is underappreciated. Second of all, the impact of literacy on the visual system is much more widespread than you would think, and it involves the primary brain regions. And third of all, the canonical view of the brain as organized according to modality is not fully complete. I would not say it's wrong, but there are more and more cases where we see it's not the whole story, and that needs to be revised. So, to wrap up, I'm just going to tell you a bit about ongoing work in our lab. So, in the story about early visual areas, we start to work with causal metals. We try to disturb the function of primary visual areas through transcranial magnetic stimulation, like the machine that has just arrived to your institution. And as far as braille reading is concerned, we have this project now where we're trying to teach braille to people who have normal visual system. Apparently, there were no people who know both braille and the visual alphabet. I'm talking about braille read with hands. When you go to a school for the blind, the teachers, they correct their pupils' textbooks by looking, the pupils' work by looking at braille. So people can read braille visually, sighted people, but there are no people, at least we couldn't find, people who can read braille with their hands and have a good visual system. So now we have a big project of teaching braille to about 30 braille teachers and also braille students, special psychology students, and we're going to compare their brains before they learned braille and after they learned braille. And also for a couple of years I have been amateur reader of behavioral economics literature, and at this point I started expanding a bit into behavioral economics, particularly in the role of associations and the role of unconscious as seen from social psychology, which is different from cognitive psychology. And since this is a school of economics, I'm basically open to suggestion of collaboration in that domain. I know how to use fMRI and how to put questions into good fMRI experiments, but I'm obviously an amateur in behavioral economics. And I want to thank you for your attention, and for those of you interested in reading, I warmly recommend the book Reading by the Brain, in the Brain by Stanislas Dehaene, and thank you very much.
So we have
for the braille readers, what their task was while they were learning in the MRI scanner?
So you're asking what was the task?
Yes.
So they were reading Braille. They were also touching like nonsense Braille, which is all 6 dots. This is not a letter. This is a nonsense character. So what you see here, basically, that activation is the contrast between Braille and the nonsense Braille, the scrambled Braille. So, for example, that contrast gives you nothing in the tactile cortex, because there you just have simple tactile activity. But they also did other things. They also did language tasks like generating verbs. So they get a noun and they have to make a verb out of that. And that actually activates other parts of the visual system. So the other parts of the visual system in the blind, they don't just sit around and do nothing, but they become a kind of a second prefrontal cortex. Suddenly they are very far away from the input and they can do memory and things like that. And that's why some people argue blind people are much better at memory tasks. But that actually activated things around here, things around V1. So this was the strongest activation in this region. Braille was the strongest activation in this region.
Okay.
You had a question, perhaps?
Oh yeah, this one is interesting, this one. But still, what kind of sample sizes do you work in your field with? Sample sizes?
Sample sizes?
Yeah.
So it depends. Like here, the results were very strong. There were just 8 subjects. In the masked recognition experiment, okay, here, for example, people see some letters very quickly, and it's masked. So you basically try to fish out that little letter that was hardly seen, that was so invisible that people miss it half the time. That gives obviously a weak signal. There you put about 20 subjects. It's between 10 and 20 subjects, depending on the task. Now with the Braille course, we're starting with 40 people, and we hope at least 15 of them learn Braille.
You mentioned that reading is mostly achieved in a parallel fashion, right? But
Yes.
how about the serial processes, like effects of letter, length in letter? Do you completely ignore it in your studies? Or?
Well, it's a bit of a simplification, but you're asking how about letter?
No, because I was wondering, like in Braille reading, you have the serial process.
Yes.
And reading is, you were saying it's parallel, so maybe like.
So
it's
I was talking about visual reading. Visual reading is mostly parallel. Let's say people now, the linguists, they are fighting whether, let's say, 4-letter word will give you 500 milliseconds, and the 8-letter word will give you 500 milliseconds or 505 milliseconds. And they're fighting about the 5 millisecond difference, but on the first approximation, it's really parallel. Up to, let's say, 9 letters where you have to move your eyes again, and that becomes longer. No, but Braille reading, of course, is serial because of the fact that you have to move your finger, and as of course you have the visual Braille that the teachers are using, but in our experience, the visual Braille is pretty slow and it's not so parallel, but you don't know, is it because they're not very good at it or because it cannot be learned? You probably know more about that in a year.
And first of all, thank you very much for the interesting talk. And I have a question regarding the scrambling.
Yes.
Did you measure the difference between differently scrambled objects or words, for example, like cut into 4 pieces or 8 pieces or 16 pieces, and how the performance decrease with scrambling?
Well, I did not do this, but there are several studies, like object recognition has been studied with scrambled objects many times. What do you mean by performance? You mean recognition or brain activity?
I mean recognition of the words, activation of the brain areas. Well, if subject understand the word, even if it's scrambled.
Yeah.
how much the performance of the subject decrease when you scramble the word more and more? Well, it's quite obvious that if you scramble the word into 4 pieces, it is quite easy to understand. But when you scramble it more and more, how much the performance decreases? Can this be trained? For example, if the subject is trained with the same task, with recognition of scrambled words?
So here we scrambled them completely and there was 0 recognition. And now what would happen if you do it slowly? My guess is that the single biggest difference between scrambled and entire words is in the higher level regions, visual from area. So it's up high in the system, and probably there, as you scramble, as long as you keep some letter shapes intact, you would still have some recognition. But in general, what we know from objects that have been studied more and more is that already the more you scramble, the less recognizable it is, the less difference you see. But of course, at some point, scrambling to 16 pieces and scrambling to 200 pieces makes no difference.
And if there is any significant change in recognition of scrambled Latin words and hieroglyphic words between different subjects, for example, like better performance in hieroglyphs because they have like very special curves and hooks and so on, and Latin words readers.
Yeah, well, I'm not sure if I understand your question, but what I can say is that if you look at Chinese, you mean hieroglyphs, you mean Chinese words, or you mean like, so logographs, actually if you take logographs, let's take them. Actually, the basic building blocks of all alphabets are very similar. You take Chinese, Egyptian, all alphabets are made of T, X type line junctions. Now in Chinese you have more features packed into single characters, but if you analyze, there is work, for example, by Marcin Szwed, who is a computational vision scientist, and he took like 60 alphabets of the world and looked at the recurrent features, and he found that the distribution of basic building blocks like T's, X's, and circles is basically almost the same, starting from alphabetic alphabets like Cyrillic or Latin, going up to logographic alphabets like Chinese. So there is a, let's say, when you scramble, you should see, if I took this stimulus and I rearrange those lines, like here, you should not see the difference, especially if here I had a lot of rounded words. But if I use words without the O's and D's, you will not be able to see the difference between scrambled Chinese and scrambled Latin.
And among scramblers, do you control for spatial frequency here somehow?
This does not. It's a good question. This destroys the spatial frequency. Actually, this has higher spatial frequency. So yes, it's not controlled. Other people, if you want to keep spatial frequency, you do Fourier scrambling, basically.
Yeah.
You do phase scrambling. This way it has, I think it's a good way to, if I'm correct, it's a way that preserves the frequency values, at least according to a Fourier transform of the visual image. But this gives a very similar, let's say, scrambled words, at least scrambled properly, will give you a similar signal in the visual system as Fourier phase scrambled words that keep the frequency. But to be honest, there is no perfect way of scrambling, and the visual, there will be lots of argues what's the perfect scrambling method, and there isn't really one.
Yeah, the reason I'm asking is because your main difference is in the early visual areas, so that spatial frequency may be important there. So the difference may be possible.
Yes, so this has higher special frequency. We control for many things. We control for number of features, for luminosity, for line length, but the scramble things naturally have higher frequency. Frequency, as you're right, is what drives early visual areas, and that's why for objects, you will see that scramble objects give you slightly more activity than regular objects because this has higher frequency. And this is something that has been seen in several labs. It's not just our experiment. So even if you have a frequency effect for scramble objects, I seem to have lost that title, but this is scramble words. So for scramble words, it's basically overwhelmed by this extra expertise-dependent activation that you have for entire words. So even if it's drowned out by other effects.
Question maybe I was left. So correct me if I'm wrong, but you said that, okay, if you look at subjects trying to comprehend spoken speech, right?
Yes.
You see specific activation in the early stream and the auditory areas, right? In the subjects of reading, you see activations in the visual areas, right?
Yes.
Has anyone looked at subjects, for example, trying to comprehend explicitly read speech?
Explicitly?
Meaning somebody is explicitly reading in front of them.
Yes.
So would that, are there differences between?
I would say that would drive a bit the visual, the word from area. Let's say there are ways to activate the reading area with with spoken language, but to do that you need to somehow bring the people's attention to the visual aspect.
Well, that's
the best way is to give them, let's say, flash a word like left, and then ask, was there a letter? No, say, play word left. So this is auditory stimulation, and then ask, does this word have an L, or does this word have a T? So it's spoken language, but it's kind of force people to reach for the orthographic knowledge.
So you should ask for silent letters.
Yes, but in the case of
laugh and ask them if there was a G.
Yes, yes. But I guess if you see somebody reading aloud, there would be maybe some imagery, like to try to impersonate. You can drive. We have no data. You can drive the visual word form area just by asking people to imagine they're reading something. It's much weaker than really reading something, but it's you can do that. Imagery drives the higher level regions, so the face region, the reading region. They are very susceptible to be activated by imagery or hypnosis.
Can I ask you a question? So you showed us a wonderful project with Chinese and French people. I just wonder, could it be that, for example, Serbian Croatian language is a better example? You have exactly the same language, two different inputs, Cyrillic or Latin. So it would be actually the better model for your hypothesis, because you have exactly the same semantics, just two different inputs.
Well, that would be a good model, but that would be a strong hypothesis. The strong hypothesis is that you would, it's enough to go from Latin to Cyrillic to lose this early visual effect. But I tried to maximize my chances, and I chose two very visually different languages because for me, Latin and Cyrillic have many letters that are in common, and they happen in the same visual space. They're left to right. So I'm not sure what will be the result. Maybe there will be some residual activity. So visual perceptual learning is a very simple thing. It's an implicit type of learning. Whatever resembles the trained thing is enhanced. So maybe expertise in Latin alphabet in Croatians would give some boost for Cyrillic activity in V1 as well, despite the fact that this is not recognized properly.
So do we have some further questions?
Might be a politically interesting result.
Yes, I would rather do that in a different country with less political Turkey, Arabic and Latin. It's we're trying actually. We had one project where we tried to get people in China, actually in Central Asian part of China, you have some types of Arabic script are left still read top down, and that gives you an opportunity to compare scripts that have where you learn in this part of the visual field to scripts in this part of visual field. But so far, this finding subjects and bring them to Shanghai, where our collaborators are, has not been very fruitful.
Because we start to be out of the time, so if we don't have some further questions, so we can